Ever wondered why we yawn all day, but dream of changing the world in a major way? It's the Bored and Ambitious Show.
Victoria Gray is a mother from Forest, Mississippi. For 34 years, she lived with sickle cell disease, a genetic condition that twists red blood cells into jagged crescents that clog blood vessels and cause agony, pain crises that sent her to the emergency room, hospitalizations that kept her from her children, the constant fear that she wouldn't live to see them grow up. In 2019, doctors extracted her blood stem cells, used a gene editing tool called CRISPR to fix a single genetic instruction, and put the cells back. Her disease is gone. She said, quote, "I feel like I have superpowers. I can do things I never thought I'd be able to do. That tool exists because of a discovery made 66 years earlier." On February 28, 1953, Francis Crick walked into the Eagle Pub in Cambridge, England, and announced to the lunchtime crowd, quote, "We have discovered the secret of life." He and James Watson had worked out the structure of DNA, the molecule that carries the instructions for building every living thing on Earth. This is the story of how we learn to read the code of life, how we learn to fix it, and the extraordinary responsibility that comes with that power. Consider this. The cost of reading a single human genome has plummeted from $2.7 billion in 2003 to under $200 today. In the span of a single human lifetime, we went from not knowing what DNA looked like to editing it letter by letter in a living patient. But to understand how a mother in Mississippi was healed by a tool discovered in bacterial immune systems, you have to go back to a monastery garden, a friar, and a question no one could answer. That traits passed from parents to children was obvious to anyone who'd ever looked at a family. But how they passed was a mystery. In the 1850s, Gregor Mendel, an Augustinian friar in Brno, began crossing pea plants in his monastery garden, tall plants with short, purple flowers with white. He counted everything, and what he found was extraordinary. Traits didn't blend like paint. They followed precise mathematical ratios. 3 tall plants for every one short. Dominant and recessive traits, governed by discrete hereditary units, what we would later call genes. Mendel published his results in 1866. Almost no one read them. His paper sat in obscurity for 35 years until 1900, when 3 scientists independently reached the same conclusions and discovered that Mendel had already been there. Genetics was born, a science with laws but no understanding of their physical basis. Thomas Hunt Morgan's fruit fly experiments proved that genes sat on chromosomes, physical structures visible under a microscope. But chromosomes contained both protein and nucleic acid. Which one carried the information? Most scientists bet on protein. It was more complex, more varied, seemingly better suited to carry the instructions for life. DNA seemed too simple. They were wrong. In 1928, Frederick Griffith showed that dead bacteria could transfer hereditary information to living ones. Something, a transforming principle, was carrying genetic instructions between cells. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty purified that transforming principle and proved it was DNA, not protein. Joshua Lederberg later said Avery gave us, quote, "the first grueling, convincing evidence that DNA is the master substance of the gene." Many scientists still couldn't believe it. Then in 1952, Alfred Hershey and Martha Chase used radioactive tracers to prove definitively that when viruses infected bacteria, it was DNA, not protein, that entered the cell and directed the infection. The final confirmation: DNA was the molecule of heredity. Whoever solved its structure would understand how life works at the molecular level. The race was on. 3 teams competed. Linus Pauling at Caltech, the greatest chemist alive, who had already solved the structure of proteins. Everyone assumed he would win. He proposed a triple helix. It was chemically impossible. At King's College London, Rosalind Franklin was producing the best X-ray images of DNA in the world. She was brilliant, she was meticulous, and she was working against institutional sexism that would have stopped most people. In May 1952, she captured Photo 51, an X-ray diffraction image so clear it revealed DNA's helical structure with stunning precision. It is one of the most important scientific images ever taken. The most important thing about Photo 51 is not the image, it's the scientist who made it. At Cambridge, James Watson, a young American biologist with no patience for caution, and Francis Crick, an older British physicist who talked constantly and brilliantly, were building models from others' data. Then came the crucial moment. Maurice Wilkins showed Watson Photo 51, Franklin's photograph, without her knowledge or permission. Watson recognized immediately what he was seeing. Was this data shared or stolen? The history of DNA cannot be told without confronting that question. In February 1953, Watson and Crick put it together, metal plates and rods representing atoms and bonds. Watson realized that adenine paired specifically with thymine and guanine with cytosine, the base pairing that explains how DNA copies itself, each strand a template for the other. Beautiful, simple, and immediately convincing. Their paper in Nature, April 25, 1953, was just one page. It contained perhaps the most understated sentence in the history of biology. Quote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Franklin and Wilkins published their X-ray data in the same issue. Franklin didn't know Watson and Crick had seen her work." She died of ovarian cancer in 1958 at age 37, likely caused by the radiation from her X-ray crystallography. She once wrote, quote, "Science and everyday life cannot and should not be separated." In 1962, Watson, Crick, and Wilkins received the Nobel Prize. Franklin was ineligible. She was dead. The controversy endures, but what she achieved endures longer. The structure was solved. Now came the harder question: how does DNA actually work, and could we learn to use it? In 1958, Crick articulated what he called the central dogma of molecular biology. DNA is transcribed into RNA, and RNA is translated into protein. Information flows in one direction, from code to molecule, but the code itself was still unbroken. DNA is written in a four-letter alphabet, A, T, G, and C. Proteins are built from 20 different amino acids. How does one language translate into the other? In 1961, Marshall Nierenberg and Heinrich Matthaei cracked the first word, 3 letters, U, coded for a single amino acid, phenylalanine. By 1966, all 64 three-letter combinations had been mapped. The entire genetic code was deciphered. And here is what stopped scientists in their tracks. The code is universal. The same three-letter words mean the same amino acids in bacteria and in humans, in oak trees and in jellyfish. Every living thing on Earth reads DNA the same way. As Crick himself marveled, quote, "The genetic code is universal. The machine that translates it is fundamentally the same in all living things. The genetic code is not a metaphor. It is literally a code, an information system 3.5 billion years old. Biology became an information science, and information, once understood, can be engineered." In 1973, Stanley Cohen and Herbert Boyer combined DNA from 2 different organisms for the first time in history, the birth of genetic engineering. Any gene could now, in principle, be inserted into any organism. The power of this was immediately obvious, and so was the danger. In February 1975, 140 scientists gathered at Asilomar, California, and did something almost unprecedented. They voluntarily paused their own research. They didn't know what they might create, so they stopped to build safety guidelines first. Paul Berg, who organized the conference, later reflected, quote, "If we stop every time we get scared, we'll never cure anything." But the pause built public trust, and it became the model for scientific self-regulation that endures today. The first payoff came fast. In 1976, Herbert Boyer co-founded Genentech, the first biotechnology company. 2 years later, Genentech scientists inserted the human insulin gene into bacteria, and those bacteria began producing human insulin. In 1982, the FDA approved Humulin, the first genetically engineered drug. Diabetics who had relied on insulin from pigs could now receive their own species insulin, produced by engineered microorganisms. Growth hormone followed. Hepatitis B vaccine, erythropoietin for anemia. Genetic engineering became routine, producing medicines that would have been impossible a generation earlier. Then came the most ambitious biology project in history. In 1990, the Human Genome Project launched, an international effort to sequence all 3.2 billion base pairs of human DNA. 15 years, $2.7 billion, laboratories on 3 continents. A public race emerged between the government project, led by Francis Collins and Celera Genomics, Craig Venter's private company. Competition accelerated progress. On June 26, 2000, President Clinton announced the draft sequence at the White House. With Collins and Venter standing together, their rivalry resolved into shared triumph. Clinton declared, quote, "Today, we are learning the language in which God created life. The genome held surprises. Humans have only about 20,000 protein-coding genes, far fewer than anyone expected. Most of our DNA doesn't code for proteins at all. Scientists initially dismissed this as junk. They were wrong again. And the transformation since has been staggering. Over 40 million people have now taken consumer genetic tests. The cost of reading a genome continues to fall. But reading was never the final goal. The goal was healing. And for that, we needed a better tool. CRISPR-Cas9 was discovered in bacteria, where it functions as a primitive immune system, remembering viruses and cutting their DNA. In 2012, Jennifer Doudna and Emmanuel Charpentier published a landmark paper showing this system could be reprogrammed to edit any gene in any organism. Doudna later said, quote, "We can now start to think about curing these diseases. That's a very different world. Earlier gene editing tools were like hammers, powerful but crude. CRISPR is like a word processor, precise, targeted, programmable. A guide, RNA, directs the Cas9 enzyme to the exact spot in the genome where you want to make a change. The cell's own repair machinery does the rest. In 2020, Doudna and Charpentier won the Nobel Prize in Chemistry, the first Nobel in Science awarded to 2 women without a male co-laureate. And by then, CRISPR was already saving lives. Victoria Gray's full story is this. In July 2019, doctors at Sarah Cannon Research Institute extracted her blood stem cells. They used CRISPR to edit the BCL11A gene, reactivating the production of fetal hemoglobin, a form of hemoglobin that doesn't sickle. After chemotherapy to make room in her bone marrow, they returned the edited cells. Her sickle cell crises stopped. Her hemoglobin normalized. Over 5 years later, she remains pain-free. She said, quote, "I'm a normal mom now. I can do things with my kids that I never thought I'd be able to do." In December 2023, the FDA approved Casgevy, the first CRISPR-based gene therapy for sickle cell disease. It costs $2.2 million per patient. Over 100 CRISPR clinical trials are now underway worldwide. But power without guardrails is dangerous. In 2018, a Chinese scientist named He Jiankui announced that he had used CRISPR to edit the genes of human embryos, creating the first gene-edited babies. The global scientific community condemned him. He was sentenced to 3 years in prison. His experiment stands as a warning about what happens when ethics fail. As Charpentier said, quote, "We need to be cautious. We need to be humble. But we should also be hopeful. CRISPR gave us the power to edit the code of life. The question now is not whether the technology works. It does. The question is whether we're wise enough to use it well. If we can edit genes that cause disease, can we also edit genes that affect height or intelligence or appearance? Where is the line between therapy and enhancement? This is a genuine dilemma, not a settled answer, and it carries a shadow. In 1927, the United States Supreme Court upheld a law allowing the forced sterilization of people the state deemed unfit. Justice Oliver Wendell Holmes wrote, quote, "3 generations of imbeciles are enough. Over 60,000 Americans were forcibly sterilized under eugenic laws. This history is not ancient. It is the permanent warning. Knowing what went wrong is how we prevent it from going wrong again. But here is what's worth noting. We are building guardrails. The Genetic Information Non-Discrimination Act, signed in 2008, prohibits discrimination based on genetic information in health insurance and employment, passed before most people had ever had their genomes read. The Asilomar tradition of scientific self-regulation continues. The global condemnation of Jiankui proved the scientific community will enforce its own ethical boundaries. As Dudna has said, quote, "The responsibility for how gene editing is used belongs to all of us, scientists, policymakers, and citizens alike." The technology is advancing faster than our ethical frameworks, but this has always been true of transformative technologies. The answer to the challenges of genetic power is more wisdom, not less science. And the science is moving. Over 50 gene and cell therapies are now FDA approved. Luxturna is curing inherited blindness for $850,000 per treatment. CAR T-cell therapy is reprogramming patients' own immune cells to fight cancer. Base editing and prime editing, the next generation of precision tools, are entering clinical trials now. But the access crisis is real. Casgevy costs $2.2 million. Sickle cell disease affects 300,000 newborns every year, mostly in sub-Saharan Africa. A cure that only the wealthy can access is an incomplete revolution. The question is not whether gene therapy works. It does. The question is who gets it. Meanwhile, the genome has become a history book. Svante Pääbo sequenced the Neanderthal genome from bones 40,000 years old. He discovered that modern non-African humans carry 1 to 4% Neanderthal DNA. Proof that our ancestors interbred with another human species tens of thousands of years ago. He won the Nobel Prize in 2022. Synthetic biology is pushing further still. Craig Venter's team created the first synthetic cell in 2010. An organism running entirely on human-designed DNA. Students in the iGEM competition are engineering bacteria to detect arsenic in drinking water. The frontier is no longer modifying life. It's designing it. Genomic medicine is becoming routine. Whole genome newborn screening pilots are underway in multiple countries. Over 400 FDA-approved drugs now carry pharmacogenomic information on their labels, your genetics determining which drug works best for you. Cancer treatments are being tailored to the mutations in your specific tumor. The technology exists. Who decides how it's used, scientists, governments, markets, individuals, is the defining question of the genetic age. Before we look forward, step back. Consider what DNA has taught us about what life actually is. Every organism on Earth uses the same genetic code, the same four-letter alphabet, the same molecular machinery to read it. Bacteria and humans, trees and jellyfish. A code 3.5 billion years old still running in every cell of every living thing. Theodosius Dobzhansky said it best. Quote, "Nothing in biology makes sense except in the light of evolution. DNA is the molecular proof. We are texts written in 4 letters, each of us a variation on themes billions of years old, and we are the first living things that can read the language we're written in. That should stop you in your tracks. We know the code, but we don't fully understand the program. How does a single cell develop into a human being? How do genes and environment interact to produce who you are? Every cell in your body carries the same DNA, yet a liver cell knows it's not a brain cell." Reading the genome was the beginning, not the end, of understanding. Decades of discovery lie ahead. Return to where we started. Victoria Gray, the mother from Mississippi. A lifetime of pain crises, hospitalizations, and fear. In 2019, scientists took cells from her blood, used CRISPR to fix a single genetic instruction, and put the cells back. Her disease is gone. She's raising her children. Her daughter asked her, "Mommy, does this mean you won't be sick anymore?" And she could finally say yes. That's not a hypothetical future. That's a Tuesday in the 21st century. The code of life was cracked by a physicist who talked too much, a biologist who was too impatient, a crystallographer who was too brilliant to be stopped by sexism, a friar who counted peas, and thousands of researchers who built on each other's work across a century. No single genius, a cumulative human project. The double helix that Watson and Crick built from metal plates is now a precision instrument in the hands of healers. The secret of life is no longer secret. The code is read, the editing has begun, and the first patients are already healed.